Hello, my name is Joshua Gabi. I welcome you to Coordination and Control. Question 1 to 5. The graph below shows the number of receptor cells, type A and type B, in arbitrary unit in the human retina along a horizontal line from the nozzle side of the eye to the outer side. Part A, giving reasons, identify the types of receptor cells presented by A and B. So when we look at the graph, we have this dotted one. That one is receptor A. Then we have this dark one here. That is receptor B. Receptor cells A represent rods. No concentration at point 6.3. No concentration of a fovea. So if we first look at receptor A, this one here, coming down here, and then moving up, and then down here, then it goes up, and then it decreases. We are saying that this one is what we are referring to as receptor cells A. There is no concentration at 6.3 and no concentration at the fovea. Receptor cell B represents the cones. Highest concentration at 6.3, the highest concentration of fovea. So for the receptor cell, which we are calling the cones, they have the highest mark here. That is at 0.6.3. That's where we have the highest mark. And therefore, that receptor cells or receptor cell is representing the cone, where the highest concentration is at 6.3, highest concentration at the fovea. Part A, Roman number 2. Explain why there are no receptor cells at position 5. Explain why there are no receptor cells at position 5. At position 5, where there are no receptor cells, is called the blind spot. Neurons from the rods and the cone pass in front of the retina, where they converge to form the optic nerve. They are so densely packed that no receptor cells occur. At point 5, if you look at it at point 5 there, you will see that at this point here, there are no receptor cell. There are no rods. Rods come here, and there is nothing at that point 5. And when you also look at the cones, they are low here, but when they reach there, they go to 0 and then they rise up here. There is no cones, no rods at that point. And that point where there are no cones and no rods, it is what we call the blind spot. Part 2. What is the name given to the region of the retina at position 6.3? Give reasons for your answer. It is the fovea. Can be called fovea centralis or the yellow spot. This one has the highest concentration of cones, and it lacks receptors A, which are the rods. And that point that we are talking about is this point at 6.3. 6.3, this point here, where we have no rods, but it is where we have the highest concentration of the cones. We call that point the fovea. Part B. Explain why the greatest concentration of receptor cell type B occurs at position 6.3. The large concentration of receptor cell type B, which are the cones, at position 6.3 is that it lies on the optical axis, directly opposite the center of the lens. It is here where the greatest refraction of light waves occur when a person is looking directly on an object. So we are saying this part here, which we have the highest concentration of receptor cells B, or what we are calling the cones, lies at the optical axis directly opposite the center of the lens, and it is where the greatest refraction of light waves occur when the person is looking directly at an object. Type B receptor cells, the cones, they are sensitive to high light intensities. Hence, their high concentration where the light intensity is the greatest. So the greatest light intensity is at the fovea. So that's where we find the highest concentration of the cone, or what we are calling type B receptor cells. Part B, explain why, Roman number 2, on entering a dimly lit room, object in the room at first are invisible, but gradually they become visible. In bright surroundings, circular muscles are contracted and the pupil is constricted. This reduces the amount of light entering the eye. It reduces overstimulation of the cone cells. On entering a dimly lit room, the radial muscles of the iris contract and the pupil slowly dilates to allow maximum amount of light to enter the eye. The process takes little time, during which so little light enters the eye that the threshold value for stimulating the rods, which are sensitive to light of low intensity, is not reached, and so nothing can be seen as the pupil dilates fully, such that the threshold value of the rods is reached and the object becomes visible. Part B, explain why in a dim light room objects are only visible in black and white color. The rods, however, do not respond to light of various wavelengths in the same way that the three types of cone cells do. For this reason, objects in the dim light room are visible only in black and white. Part C, Roman 1. From the graph, identify and describe the features of the receptor cells which allow color vision. Highly concentrated at 6.3, low concentration at 5, evenly distributed between 1 to 4.8 and 7 to 10. Increases rapidly between 6 to 6.3 and decreases rapidly between 6.3 to 7. Part C, Roman 1. The flowers of 3 species of the plant are similar in form and appear to be having yellow color of petals when photographed in ultraviolet light. Each species shows a different pattern on its petal. Using this information, explain how the bees are able to distinguish between the flowers of the three species, but not humans. Ultraviolet light comprises of ranges of wavelength in much the same way that visible light does. The corn cells of humans cannot distinguish different ultraviolet wavelength, and the light from the petal stimulates the red and green cones uniformly, giving appearance of yellow. In bees, rhabdom and their compound eyes can distinguish the different wavelengths of ultraviolet light. The petals must have a pattern of pigment that reflects the different ultraviolet light differently. The different wavelengths reflected produce a pattern when perceived by bees. In each species, this pattern must vary, allowing the bees to distinguish the wavelength. Number 2. The theory of color vision suggests that the photoreceptor has pigments that exist in 3 forms, namely red, blue, and green, according to the color of wavelength absorbed by each. The absorption of different wavelengths by the 3 forms of photoreceptor pigments is given in the table below. Study the information given and answer the questions that follow. So we have different wavelengths, and we have the amount of light absorbed as a percentage of maximum. We have the red cones, the green cones, and then we have the blue cones, and the amount of wavelength they absorb. Part A. From the data, explain why light of wavelength 430 nanometers appear blue. If we look at the light of 430 nanometer, you will see that it is mainly absorbed by the blue cones. 0 red, 0 green cones, but 100% blue. So in the 430 nanometer wavelength, it shows no light absorbed by the red and the green cones. If you look at the red, they don't absorb any light of that wavelength, and even the green cones don't absorb that light. Whereas the blue cones, they absorb the maximum amount of light at 100%. So if you look at the blue cones, they absorb 100%. Consequently, only the blue cones are stimulated by the light of 430 nanometer, which gives the sensation of the blue light. From the data, explain why light of wavelength 550 nanometers appears yellow. So when we look at 550 nanometer, this light appears yellow. But you can see that the cones, they absorb 85%, and the green cones also absorb 85%, whereas the blue cones, they absorb 0%. So in the 550 nanometer, there is no stimulation of the blue cones, but the red and green cones are equally stimulated, which the brain interprets as yellow. No stimulation by the blue cones because there is no absorption of that light, and there is equal stimulation of the green and red cones. So the brain interprets that one as yellow. From the data, explain why light wavelength of 570 nanometer appears orange. We look at the wavelength of 570 nanometer. They say it appears orange. And you can see that the red cones, they absorb 100% of that light, and the green cones, they absorb 45% of 570 nanometer wavelength light. But the blue cones, they don't absorb any of that light wavelength. The red cones have the maximum absorption, which is 100%. And the green cones, they are at 45% of this wavelength. Whereas the blue cones, again, absorb no light in that wavelength. So the brain interprets the impulse received predominantly red, with some green included. Such a mixture is as orange. Part B. Explain why two closely placed small objects can easily be distinguished by the cones than rods. Each cone has its own separate neuron connecting to the brain. Whereas groups of rod cells share a neuron. Therefore, light from such objects will enter the eye as two parallel beams very close together, show retinal convergence. If these stimulate adjacent cone cells, two separate objects are interpreted, which we call visual acuity. If, however, two adjacent rod cells are stimulated, the chances are that they will be connected to the same neuron. This simultaneous stimulation of the two rod cells therefore results in a single neuron, sending impulses to the brain, which will be interpreted as a single object. That is, cannot distinguish the two objects. Number 3. An investigation was carried out on the effect of applying different concentrations of auxins on roots and shoots of oat seedlings. Results in table 1 were obtained, were expressed as percentage stimulation, positive, or inhibition, negative, of growth, compared with untreated controls. Use the information to answer the questions that follow. So we have our concentration of auxins applied, parts per million. Present the results in a suitable graphical form. So we are having our x-axis, which is going to represent the auxin concentration in parts per million, and we have the y-axis, which is representing the percentage stimulation of growth. Part A, Roman number 2. Using the graph, describe the effect of different concentrations of auxin on the root and the shoot growth. We shall start with the shoot. The concentration between power -6 to power -4 has no effect on the growth. So if you look at the concentration of auxins applied on the shoot from -6 to around -4 here, you can see that concentration has no effect on the growth of the shoot. And between 10 to power negative 4 to 10 to power negative 2, growth increases slowly. So from negative 4 to negative 2, you can see that there is an increase in the slow growth of the shoot. And an increase to the peak at 100. Rapid increase up to the 100. So when you look at it from 10 to power negative 2 to 100 parts per million of oxygen concentration, there is a rapid increase up to the peak growth in the shoot. And then afterwards, it drops to the concentration of 10 to power 2. So from there, we can see that the growth rate decreases as the concentration increases to 10 to power 2. Using the graph, describe the effect of the different concentration of oxygens on the root and the shoot growth. Now we want to see the effect on the root. There is a slow increase in growth rate between 10 to power negative 6 and up to 10 to power negative 5. So when you look at that concentration from 10 to power negative 6 up to 10 to power negative 5, there is a slight increase in growth of the root and a constant rate between 10 to power negative 5 up to 10 to power negative 4. So from 10 to power negative 5 to 10 to power negative 4, there was a constant growth rate of the root from 10 to power negative 5 up to 10 to power negative 4. Then the rate declines first to 100. So it declined first to 100. When you look at it, it declined very fast to 100. And slowly, rapidly to 2 to 110 to power 2. So it declined very fast to 10 to power 2. Here, declined very fast to 10 to power 2 here. Part A, Roman number 3. From your graph under table 1, point out the differences between the responses of the roots and shoots to the different concentrations of oxygens. Very low concentration of oxygen, that is from 10 to power 6, negative 6 to 10 to power negative 4 parts per million. From 10 to power 6 to 10 to power 4 parts per million, it stimulates the root growth while there is no effect on the shoot. So you can see from here to here, there is a constant growth rate in the shoot. Whereas from the same, you see there is an increase in the growth rate of the root. At higher concentration of oxygens, that is from 10 power negative 4 to 10 to power 3 parts per million, there is a decrease or inhibits root growth. While from 10 to power negative 4 to 100 parts per million, it increases or stimulates the shoot growth. So we can see from 10 to power negative 4, it inhibits the growth of the root. Whereas from 10 to power negative 4, it stimulates the growth of the shoot. Increase in oxygen concentration rapidly inhibits the shoot growth, whereas it slowly inhibits the root growth. So when we increase the concentration of oxygen, it rapidly stimulates the shoot growth and slowly it inhibits the root growth. Figure 1 shows the changes in the membrane potential, showing electrical events associated with the nerve impulse in axon, while Figure 2 shows the changes in permeability of the membrane of the axon, sodium and potassium ions during the transmission of an impulse which occurs very fast and rapidly. Compare the trend of diffusion of sodium and potassium ions across the membrane of an axon over a 3 millisecond period. We shall look at the similarities. Here we have our graph of the diffusion of sodium and potassium ions per given time. We can see in both the diffusion of sodium and potassium ions, they increase rapidly to the maximum. So when you look at sodium, sodium increases rapidly up to the maximum. And when you also look at that one of potassium, it also increases rapidly to the maximum. So in both diffusion of sodium, steel, and potassium ions, they reach the maximum peak. You can see that all of them, they reach the maximum peak. Sodium reaches the maximum peak. And then you can also see that also potassium reaches the maximum peak. In both sodium ions and potassium ions, the diffusion declines rapidly after the peak. So you can see that for sodium, after the peak, it declines very rapidly. And also, you can see that for potassium ions after the peak, it also declines very rapidly. In both the diffusion of sodium and potassium ions, it begins to increase from 0.2. So both of them are starting to increase from 0.2 millisecond. You can see that all of them, they will start from 0.2 to increase. Sodium increases from 0.2 and potassium also start increasing from 0.2 milliseconds. In both the diffusion of sodium ions and potassium ion is the same at 1 millisecond. At the point of 1 millisecond, around here, both of them, they are the same. The rate of diffusion becomes the same at 0.1 millisecond. Let's now look at the differences. The diffusion of sodium ions are higher, while those ones of potassium ions are lower, between 0.4 milliseconds and 1. So when you look between 0.4 millisecond around here and 1.2 around here, the sodium ions are higher. Sodium ions are higher, and the potassium ions, you can see that during that period they are lower. Potassium ions during that period, they are lower compared to the potassium ions. So potassium ions are lower compared to the sodium ions. And the diffusion of sodium ions is lower, while the diffusion of potassium ions is higher, from 1 to 1.2 milliseconds. So when you look at between 1 and 1.2, between 1 and 1.2, you can see that the potassium ions are higher, whereas the sodium ions are lower. So between 1.2 and 1 milliseconds, the diffusion of sodium ions is lower compared to the diffusion of potassium ions. The diffusion of sodium ions decreases rapidly, while the diffusion of potassium ion increases rapidly between 0.8 to 1.4. So from 0.8, the sodium ions will decrease rapidly up to 1.4, whereas during that same period, the potassium ions increases very rapidly. The diffusion of sodium ions increases more rapidly, and the diffusion of potassium ions decreases from 0.4 to 0.8. So from 0.4, roughly here, to 0.8, the diffusion of sodium ions increases rapidly than the diffusion of potassium ions. You can see that the diffusion of potassium ions is increasing slowly. The maximum diffusion of sodium ions was reached at an earlier time, whereas the maximum diffusion of potassium ion was reached at a later time, around 1.5. So when you look at the maximum diffusion of sodium ions, it was reached at a lower time, whereas the maximum diffusion of potassium ions was reached at a later time. The diffusion of potassium increases between 1.3 to 1.5, 1.3 to 1.5, and declined rapidly to 3 milliseconds, while the diffusion of sodium ceases. So from 1.3, the diffusion of potassium increased rapidly, and then from 1.5 it decreased rapidly, whereas the one for sodium, it ceases by that time. The diffusion is 0. Part B. Use both figure 1 and 2 to explain the trend of each of the following during propagation of the impulse in the oxygen, the membrane potential. Between 0 and 0.4 milliseconds, the negative membrane potential is kept constant at negative 60 millivoltage. So when you look from 0 to 0.4, then membrane potential is maintained at negative 60 millivoltage. This is the resting potential. The outside of the membrane of the oxygen is more positive, while the inside is more negative. So we say the membrane is polarized. Between 0.4 millisecond to 0.6 millisecond, the negative potential differences decreased rapidly to 0, and the potential difference rapidly becoming less negative. So from 0.4 to 0.6, the negative potential difference decreased rapidly to 0, and the potential difference rapidly became less negative. This is because the outside of the membrane is building a slightly negative charge, while the inside is building a slightly positive charge. Between 0.6 to 0.85 milliseconds, the positive potential difference increased rapidly to reach the peak at plus positive 40 millivoltage. So from 0.6 here, the positive potential increased rapidly to a peak of 40. Increased rapidly to a peak of 40. That is from 0.6 millisecond here. It attained a plus 40 millivoltage. This is because the outside of the membrane has attained a more negative charge, while the inside has attained a more positive charges. Membrane is now more depolarized. A threshold value is attained at the peak, resulting into the action potential. transmission of impulse. So at the peak here, we attain the action potential. The threshold value has been attained, and there is the transmission of the impulse. And between 0.85 milliseconds to 1 millisecond, the positive potential difference declined rapidly from 0.8 millisecond to 1 millisecond. There was a rapid decline in the positive potential. This is because the outside of the membrane is once again becoming slightly positive, while the inside is becoming slightly negative. Now we say the membrane is slightly repolarized. Between 1 millisecond and 1.2 milliseconds, the negative potential difference increased rapidly to 60 millivolt, the resting potential. So from 1 to 1.2, there was a negative potential difference increasing rapidly to negative 60 millivolt, the resting potential. The potential rapidly became more negative. The outside of the membrane is more positive, while the inside is more negative. Then we say the membrane is repolarized. Between 1.2 to 1.8, the negative potential difference increased beyond the negative resting potential. So from here, 1.2 to 1.8, the negative resting potential has reached beyond the resting potential. The potential difference is more negative than the resting potential. So we can see that the negative potential is below the resting potential. And this is because the membrane is hyperpolarized. Between 2 milliseconds to 4 milliseconds, the negative potential difference decreases gradually. The potential difference becomes gradually less negative to attain the resting potential. The membrane of the oxygen is fully repolarized. So from 2 to 4 milliseconds, there is a gradual less negative, and we go back and attain the resting potential. So we say that at this point, the oxygen is fully repolarized after attaining the resting potential millivoltage. Let's talk about the sodium ions. Between 0 to 0.3 milliseconds, sodium ions remained constant. That is from 0 to 0.3. Sodium ions remained constant, and this was because there was net diffusion of sodium ions. The sodium gates are closed, and the protein channel specific to sodium ions are still closed. So we can see from 0 to around 0.3, sodium ions remained constant because of the net diffusion of sodium ions, as the sodium gates and protein channels specific to ions of sodium were still closed. Between 0.3 to 0.8 milliseconds, diffusion of sodium ions increases more rapidly. There is stimulus. The sodium gates and the protein channels specific to sodium ions open, so the sodium ions diffuse rapidly inside the membrane. So we are seeing from 0.3 to 0.8, there was a rapid increase in the sodium ions. This is because during that time, the sodium gates and the protein channels specific to sodium ions were opened, which caused a rapid diffusion of sodium ions inside the membrane. Then between 0.8 to 1.2 milliseconds, sodium ion diffusion declined rapidly. And this is because an impulse had already been transmitted and the sodium gates closed. So when you look from 0.8 to 1.2, there was a rapid decline in the diffusion of sodium ions. This is because the impulse had already passed or been transmitted, and therefore the sodium gates were closed. Now let us look at the potassium ions. From 0.4 milliseconds to 1.5, the potassium ion diffusion increased rapidly. The action potential has already been achieved, and many protein channels specific to potassium ions open. So the potassium ions diffused rapidly outside the membrane. So we can see from 0.4 to 1.5, there was a rapid diffusion of potassium ions because the action potential had been already achieved and many potassium protein channels specific to potassium ions opened, thereby potassium ions diffusing rapidly outside the membrane. Between 1.5 to 3 milliseconds, the potassium ions diffusion declined rapidly. The membrane is become impermeable to outward diffusion of potassium ions, and the protein channel specific to potassium ions close, and potassium ions do not diffuse rapidly outside. So that is from 1.5 to 3. The potassium ion diffusion rapidly declined because the membrane now becomes impermeable to the outward diffusion of potassium ions, as the protein channels of potassium ions specific to potassium ions closes, and therefore the diffusion of potassium ions outside are reduced. Part C. In each case, state two factors which can cause the rapid and slow propagation of impulses. Let's start with the rapid propagation of impulses. This can be caused by having a myelination of the membrane of an axon. That will increase rapid propagation of the impulse. If there is a large diameter of the axon, also increase the rapid propagation of impulses. If there is high body temperature or the endotherm, will increase the impulse transmission. High concentration of sodium and potassium ions in the body can cause a rapid propagation of impulses. What of the slow propagation of impulse? What causes the slow propagation of the impulse? If there is non-myelination of the membrane of the axon, if the membrane of the axon diameter is smaller, lower body temperatures, and low concentration of mineral ions. All those ones will slow the propagation of the impulses. Part D, give the significance of fast conduction of impulses to an organism. Quick and fast detection. It helps it to escape response from the predators or dangers. Fast conduction, there is fast detection of the sudden changes in the external and the internal environment. Homeostatic control mechanism, they are achieved fast and easily. There is fast stimulation of the glands to secrete the digestive enzymes, resulting into fast digestion. There is fast response due to fast secretion of hormones. In an experiment was carried out to investigate the effect of oxine iodoacetic acid and gibberellic acid on the elongation of the segment of P stem. A control group of P segments received unadded iodoacetic acid or gibberellic acid. Other groups of P segments were treated with equivalent quantities of iodoacetic acid only, gibberellic acid only, or both iodoacetic acid and gibberellic acid. The results are shown on the graph below. Part A. Describe the effect of each of the following plant hormones on the mean stem segment elongation. First one is gibberellic acid. Increases the mean segment elongation slightly rapidly, increasing the mean stem elongation in the 24 hours and later gradually. So when you look at gibberellic acid only, which is this one here, our curve here. First of all, it increases slightly rapidly up to the 24-hour, and later it increases slightly gradually later on. Two, let's look at iodoacetic acid. This one increases the mean stem segment elongation greatly or rapidly, increasing within the 24 hours and later gradually increasing. So when you look at our iodoacetic acid, you can see that it increases the mean stem segment elongation rapidly in the first 24 hours. And then later on, it increases it gradually. Part B, Roman number 1. With reason, state the biological relationship exhibited by gibberellic and iodoacetic acid synergism. Their combined effect is much greater than the sum of their separate effect. So when you look at gibberellic acid and iodoacetic acid, when they are put together, their effect is greater than the sum of their combined effect. Part B, Roman number 2. Explain the difference in the effect on the mean stem segment elongation when the pea plant were treated with iodoacetic acid only, and when treated with a mixture of iodoacetic acid and gibberellic acid. When a mixture of iodoacetic acid and gibberellic acid has a greater effect on the mean stem segment elongation than when iodoacetic acid was used alone. So when we see the iodoacetic acid being used alone, that is what we got, the graph. But when there was a combined effect of iodoacetic acid and gibberellic acid, you can see there was a great elongation of the stem. In the mixture of gibberellic acid initiates the formation of iodoacetic acid. That is in the mixture. When there is a mixture of gibberellic acid in iodoacetic acid, this one, gibberellic acid, initiates the formation of iodoacetic acid. Therefore, more iodoacetic acid is secreted in addition to the already added, thus weakening the cell walls faster, allowing a greater elongation of the stem together. So when there is a combined effect of gibberellic acid and iodoacetic acid, iodoacetic acid has an effect of weakening the cell walls, which causes the faster and greater elongation of the stem together. And when it is put with gibberellic acid has the effect of causing further initial formation of iodoacetic acid. So we shall have more iodoacetic acid which is going to cause further weakening of more cell walls and therefore faster, greater elongation of the stem in the combination. Part C. Describe the role of gibberellic acid in seed germination. Once released by the hydrated embryo following imbibition and in consequent translocation to aleuron layer of the seed, gibberellic acid hydrolytic enzyme like the carbohydrases, like the amylase, protease, lipase, which on being transported to the food reserves catalyzes hydrolysis of the stored food substances to suitable food substances. The suitable food substances are then translocated to the embryo for respiration to provide energy for germination. Amino acids from protein hydrolysis, they are used to synthesize enzymes and tissues. Lipids are from formation of other growth substances in the embryo, and which brings about seed germination. Part D. How have farmers used iodoacetic acid to their benefits? They have used it as a selective weed killer. There is induction of fruiting in absence of pollination, that is parthenocarpy. It is used in storage of potatoes or other crops since they inhibit sprouting and prolong seed dormancy. In synthetic auxin, utilized as rooting agent in the stem cuttings and for the development of adventitious roots. The auxins are utilized as anti-abscissation agent, these preventing premature leaf and fruit fall. Auxins have been used to delay flowering until the appropriate time. Auxins have been used to increase the fruit size. Auxins are utilized as anti-sprouting agents in potatoes, prolonging their storage time. Thank you very much for participating in coordination and control. Question 1 up to question 5. Please do like, share, and subscribe.